Hello, students. This is Dr. Faizan Mirza. This question that I'm discussing here is an assessment objective 2 question that came in October-November 2025, variant 4. This question deals with small interfering RNA, something which is very unique, and you study that later at university level, or if you're interested in research, then you may come across this molecule at A level as well. Otherwise, this question at AS level sounds a bit at times confusing to the students. It may sound confusing and intimidating at the same time as well. So, what to do when such a question comes? Just don't ignore the core that you have. The core knowledge that you have is what will help you navigate through such difficult questions. So let's start with this question deals with that it says that there is a small interfering RNA, siRNA, in short length of a double-stranded RNA. So this is a type of a RNA that is double-stranded RNA, dsRNA. It's having about 21 to 25 nucleotides. siRNA helps to regulate protein synthesis. Figure 3.1 an outline summary of one of the way in which the primary transcript can be processed to produce a molecule of siRNA. So this siRNA actually comes from the primary transcript only. The transcript does not code for the amino acids. So this transcript gets made using a template DNA, and the primary transcript starts to turn in a way that it becomes siRNA rather than become a messenger RNA because it's not going to code for any protein synthesis. Step 1, just go through the details very carefully of the provided in the diagram. Step 1, they are saying there's a primary transcript which is synthesized from one strand of DNA. We know what a primary transcript is. It's always from a templated strand. Step 2, the primary transcript loops and a section of RNA becomes double stranded. So you can see that there's a 5' end, this thing, and there's a 3' end. And it does this molecule folds over itself. When it folds over itself, this region is including the section of paired nucleotides. For example, if there is A here, there will be U here. If there is G here, it's C here. So complementary base pairing can be expected here with hydrogen bonding stabilizing this part. What about this loop? This loop is pretty much similar to the loops that are there in tRNA, the loops with unpaired nucleotides. So whenever there are unpaired nucleotides and the molecule is looping, it loops in such a way that the unpaired nucleotides go this way, whereas the paired ones, they start to face one another. You can recall something of tRNA structure from there as well. Step 3, they are saying that then there is an enzyme called DROSHA that attaches and cleaves. Cleaves mean cuts. So you can use this term in the answer as well. The RNA to produce a shorter length. So this DROSHA is attaching somewhere here. And when it is removing the or cutting the end, you can see that it's cutting the DNA in such a way that these regions are not exactly one on top of the other. It's almost one nucleotide apart. Rather than cutting it on a straight line, it is cutting one strand at here and one strand at one nucleotide apart. Then similarly, in step 4, they are saying that this is being cut off, leaving behind this molecule. Then another enzyme in cytoplasm called dicer attaches and cleaves the RNA further. So it attaches here, it's at a different site and cuts this DNA again in the same way, but at a different site. This leaves the siRNA. They are saying that step 5 siRNA, which is double stranded and has two unpaired nucleotides at each end, is produced. So this is an unpaired nucleotide here, this is an unpaired nucleotide here. You have a double stranded RNA being produced called siRNA. How many nucleotides are there? There are 21 nucleotides here. Now, the questions will always be based on your core knowledge, how you can recall and reconnect the core knowledge that you already have with the new situation. Again, in this case, this is what we'll be doing. In step 2, in figure 3.1, the single stranded primary transcript loops and forms a section where dsRNA is present. Explain how is it possible for the double stranded section of RNA to be held together in step 2 and maintained this way in step 3, 4, and 5. So how this double-stranded region can be maintained? Obviously by complementary base pair. So all we'll do is just list down the details regarding complementary base pairing on the RNA, that there are complementary bases of adenine uracil facing one another, the guanine cytosine facing one another, held together by 2 and 3 hydrogen bonds respectively. And since it's an RNA, it would lack the adenine nucleotide. Let's move on. After step 3, the shorter dsRNA produced by the action of DROSHA is transported to the cytoplasm, which means DROSHA works before it's coming to the cytoplasm. which is cleaved further by dicer to produce siRNA. So that's why two different enzymes, DROSHA and dicer, are needed to cut dsRNA into shorter lengths. So why two different enzymes? Number one, because both of them will work on a different site. They have a different specific site at which they act, because their active sites will be of different shapes. Plus, both these enzymes are in different locations inside the cell. It's already told to us that dicer is working in the cytoplasm and DROSHA is working before that. So DROSHA and dicer are definitely working at two different pHs, at two different mediums, at two different locations in the cell. And DROSHA must have worked before, only then dicer can work, because the probably DROSHA enzyme is working inside the nucleus and dicer is working in the cytoplasm. Dicer is probably large in size. It cannot enter the nucleus through the nuclear pore. That's why it must wait in the cytoplasm for this RNA to come out. From 2018, siRNA have been used in therapeutic drug treatment to a number of diseases. The presence of molecules siRNA in cytoplasm can result in the cleavage of messenger RNA, molecules coding for protein involved in diseases. This prevents the synthesis of protein. Describe the differences between a molecule of mRNA and a molecule of siRNA. So again, here you are told that this siRNA can stop protein synthesis at the level of the mRNA, and the mRNA will be formed, but the abnormal protein will not be formed. So how this siRNA can work? This is the next part. Right now we are working where they are asking you to compare and just give the differences between a molecule of an mRNA and siRNA. So we know mRNA is a single stranded, where siRNA is double stranded. mRNA can code for protein, while siRNA cannot. And mRNA is much longer, way longer than siRNA, because siRNA is just 21 base pairs long. So siRNA is having anti-parallel strand as well as the strand 523 and 325 and both. Whereas messenger RNA is having only the 523 strand. Here comes another intimidating figure with a lot of detail. So let's review this figure. They are saying figure 3.2 summarizes how mRNA coding for the protein involved in the disease can be targeted and cleaved by siRNA. So how the mRNA, which eventually will code for an abnormal protein, can get like removed using siRNA. So the presence of siRNA activates a protein called AGO2, which binds to siRNA and other proteins to form a complex called RISC. So how is the RISC form? This is the siRNA. siRNA had two strands. We just studied that. There's a passenger strand given to us. This is a guide strand. They are saying that the guide strand is required for silencing the mRNA or for cleaving the mRNA, where the passenger strand is not required. Then there is an AGO2, a protein, and they are saying there are multiple proteins other than AGO2 as well. They all will bind to this molecule to form the complex called RISC. So this is what RISC is, RISC. So this RISC molecule forms with the guide RNA being there, the guide strand being there, and the AGO2 and all the proteins being assembled here. They attach at this one, and the passenger strand is released. This passenger strand already told to us is not required. So this moves out, leaving behind the guide strand. Why the guide strand need to be removed from the passenger strand? Because now the guide strand is single strand. It's a single strand molecule now. And this single strand molecule can now form complementary base pairing or hydrogen bond with messenger RNA, and then can produce a cut, thereby cutting the mRNA and making sure that its protein is not made. Moving on to the next part. With reference to figure 3.2 state why passenger strand need to be separated from released from RISC. Because RISC must be single strand of RNA to allow it to align with it and attach to the target mRNA, and to release the release of this passenger strand allow the guide R strand to get exposed. The bases can then come in contact with the mRNA, and this guide RNA will have complementary sequence to the mRNA. The last part, the aim of siRNA therapy is to prevent this thing that the disease doesn't occur. A target mRNA molecule can be cleaved in different locations. It means if there's a long mRNA molecule, it may be cut here, it may be cut here, it may be cut here. So every way, a different way, a protein synthesis will be affected. So this is how cleaving mRNA in different locations will have different effects on the protein synthesis, and explain how these different effects can result in lack of the functioning protein. So if RISC cleaves the mRNA at different locations and resulting in cleaved fragments, they can have different effects on protein synthesis. How? It can degrade the mRNA before it attaches to ribosomes, so the protein will just not be translated. It just will not be made. It may cleave the mRNA. When it leaves the mRNA at the location at the start codon, then the translation will just not begin. Or it can cleave the mRNA, ends up making abnormal protein. The cleaved fragment ends up making the abnormal protein that does not fold into a proper 3D shape, and so it gets degraded in the cytoplasm, or the cleaved mRNA produces a much shorter protein, which is then unable to carry out any function. So it's a 10-mark question, and on surface, this question looks to be quite scary, but it's not. The idea is that eventually they are just testing your in the knowledge that you have regarding protein synthesis, regarding mRNA, the complementary base pairing, the sites within the cell, and pretty much that's it. So, yes, these are assessment objective 2 questions in which you are provided with a novel situation, a new situation, and you are expected to just solve that problem using the information that you already have. I hope this video was helpful for you. That's it from my side. Thank you so much.